Thanks very much indeed, Camilla, and to everyone for inviting me. It's a real pleasure and a privilege to be here. So I'd like to try in a pretty short amount of time to give an overview of this really exciting new non-invasive technique for neuromodulation, a technique that offers much better, I think, spatial resolution than other non-invasive brain stimulation techniques, as well as the ability to target deep brain regions. And I'd also briefly like to describe our own recent forays into this field, work that has been essentially interdisciplinary with my colleagues in biomedical engineering in Oxford, Robin Cleveland and Joe Blackmore, as well as Verena Braun, who's a postdoc in my group. So first of all, just some ultrasound basics. The human hearing range lies between approximately 20 hertz and 20 kilohertz. And ultrasound is anything above the human hearing range. So dogs can hear up to about 50 kilohertz, bats up to 200 kilohertz, and the frequencies that we use for neuromodulation lie just above that, from 250 kilohertz up to about 2000 kilohertz, but lower than the very high frequencies that are used for diagnostic ultrasound imaging. So in the transducers that we use, ultrasound waves are both produced by and detected by piezoelectric crystals. These are dipolar structures, such as quartz, which deform when an electric current is applied across them. And they can be made to vibrate at high frequency by alternating current. But the effect also works the other way around, in that if compressed or stretched, for example, by an incident sound wave, a voltage is generated across the opposing surfaces. So this effect has obviously been used for a long time in ultrasound imaging, in which the ultrasound transducer emits a very brief ultrasound pulse. And then different tissue types in the body have different acoustic impedances according to their density. And at the boundary between two different tissue types, a certain amount of acoustic energy is reflected, and that can then be detected by the same transducer. And the time taken for the wave to travel back to the transducer is used to determine its distance. Now, as we'll see, bone has a very high acoustic impedance, so ultrasound is much better for imaging soft tissues, such as babies. Now, using curved transducers, such as shown here, ultrasound can be focused at very high energy levels into intracorporeal targets. So this is called high intensity focused ultrasound, or HIFU. And this technique was pioneered back in the 1950s by the aptly named Fry brothers at the University of Illinois. So HIFU has a tissue heating effect, which can be used for thermal ablation. And there's now a wide range of applications of this non-invasive technique for the treatment of tumors in prostate, in bone, pancreas, and liver, amongst other things. Now, focused ultrasound also has a mechanical effect on tissue, and the shock waves that you can generate can be used to break up things like renal stones or gallstones. Now, because of the problem that I mentioned earlier, that bone reflects and absorbs ultrasound energy so much, the use of ultrasound for intracranial applications has been very limited. However, in the last few years, arrays of over 1,000 transducers within an MR-compatible helmet have been used to focus ultrasound deep inside the brain, particularly in the thalamus, and cause tiny thermal lesions for the treatment of things particularly essential tremor. The temperature rise of up to around 60 to 65 degrees Celsius is monitored by MR thermometry, and the lesion sizes are tiny, in the order of 1 or 2 millimeters. Its efficacy and relatively few serious side effects mean that this is now routine clinical practice for essential tremor in the United States, in Canada, and a variety of other countries. And clinical trials are underway for its use in other disorders. However, it's much lower intensity of ultrasound that we're interested in for neuromodulation. Going back to the Fry brothers, they showed in 1958 that a few seconds of a much lower intensity focused ultrasound delivered to the LGN of the thalamus in the cat through a craniotomy window could temporarily attenuate the visual evoked EEG response to a flash of light. More recently, The neuromodulatory effects of ultrasound have been demonstrated in a range of different animal models, including rodents, rabbits, sheep, pigs, and non-human primates. And these movies on the right-hand side from Seung-Shik Yu at Harvard, for example, show highly focal motor responses in the forepaw above and the tail below of the rat to pulsed ultrasound. But despite its enormous potential for research and clinical applications, so far really only a handful of neuromodulation studies have been conducted in humans. These have targeted brain structures in predominantly primary sensory, primary motor and primary visual cortex, as well as a couple of deeper structures such as the thalamus and the head of caudate. And I'll just highlight 3 examples here. So firstly, Win Legon, who was at the University of Virginia at the time, showed that a 500 kilohertz transcranial low intensity ultrasound beam with a lateral resolution of about 5 millimeters and an axial resolution of about 18 millimeters, targeted to primary sensory cortex, can suppress EEG evoked potential, EEG evoked activity here to a touch on the skin of the hand, and also could produce a behavioural improvement in 2-point discrimination on the fingers and frequency discrimination. Secondly, Seung-Shik Yu and his colleagues at Harvard stimulated primary visual cortex in combination with fMRI and provoked a sensation of phosphenes in the majority of participants, as well as activation within visual pathways, as shown here, which was detectable at a single subject level. Now, motor effects seem more difficult in the few studies that have been done to demonstrate in humans with TUS compared to, say, with TMS. However, Win Legon's group, using concurrent ultrasound stimulation and TMS to primary motor cortex, showed that TUS inhibits the amplitude of single pulse motor evoked potentials, and in fact facilitates reaction time on a simple stimulus response task. But so far, no quantitative behavioral or physiological modulation of, for example, higher order perceptual pathways, cognitive pathways, has been demonstrated. It's important to understand the way in which acoustic energy is typically delivered in a TUS experiment and some of the terminology used. So I'll just cover some of that here. The ultrasound carrier or base frequency is typically between 250 and 1000 kilohertz and is delivered in a short pulse, which lasts normally about 0.5 milliseconds. Higher carrier frequencies have greater spatial resolution within the head, but they also have greater attenuation and distortion by the skull. Pulses are delivered in a train at a pulse repetition frequency of about 0.5 to 1 kilohertz. And those last for a variable length of time, typically in the order of about a few hundred milliseconds. And that's called a burst, one trial in the experiment. The burst duty cycle is then the percentage of the burst for which the ultrasound is on rather than off. The pressure of the ultrasound wave in TUS is typically less than 1 megapascal, and the intensity, where intensity is directly proportional to the square of the pressure, but inversely proportional to the density of the material through which it's traveling, is between 0.5 and 50 watts per centimeter squared. Okay, so what about mechanisms? A question we're often asked and can rarely answer. TUS has been shown, however, to directly stimulate action potentials and synaptic transmission in hippocampal slices, partially mediated through the activation of sodium ion channels and also blocked by the use of tetrodotoxin, which is a sodium channel blocker. Similar effects have also been shown for calcium channels, but how are these action potentials generated is the key question. So in contrast to the heating effect of HIFU, which I talked about earlier, which uses intensities of over 200 watts per centimeter squared, low intensity neuromodulation leads to minimal tissue heating, much less than 0.1 degrees centigrade. So thermal effects are generally not thought to be involved. Indeed, in a C. elegans model, if you knock out thermosensitive ion channels, there's no alteration in the behavioural responses to focused ultrasound. Whereas if you knock out mechanosensitive ion channels, you abolish this response. So therefore, a currently popular hypothesis is that acoustic radiation forces send shear waves through or along rather than neuronal membrane, and these either open up mechanosensitive ion channels, and that sort of increases the conductivity of the membrane, or they generate capacitance currents by stretching the membrane and build up charge across it, much like the sort of pressure phosphenes you see when you press against your closed eyelid. An alternative hypothesis shown at the bottom here is actually that ultrasound generates intramembrane cavitation, which are small regions within the phospholipid bilayer called sonophores that expand and contract, again, generating capacitance currents and build up charge, which eventually results in action potentials. But that said, a number of other hypotheses are out there, and much further investigation is needed for us to fully understand the mechanisms. But another really important thing towards broader testing in humans is firmly establishing the safety profile of TUS. The sort of parameters that people talk about are the intensity, which I've already mentioned, the maximum pressure or intensity, either during a single burst or across a period of time. And other measures are the mechanical index shown here, which is an estimate of the likelihood of that cavitation that I talked about earlier, which if it's too strong can potentially cause tissue damage, and the thermal index, which is the steady state temperature rise during sonication. There are no published safety guidelines yet for TUS, according to these measures, but so therefore we tend to use and stay well within the limits set by the FDA for diagnostic ultrasound imaging, as demonstrated by these red lines. But TUS is rather different from imaging in some ways. For example, the frequency range which is used is lower, as I've said already, and the pulse bursts are longer. So how translatable these safety limits are is debatable. However, nevertheless, despite that, recent comprehensive safety reviews of the literature, both from our group here and from Cristina Pasquinelli, have emphasized the absence of evidence of damage so far across greater than 50 animal studies. But clearly there's a lot more work to be done to establish a solid safety framework. Another concern that's arisen recently is that some ultrasound effects in live animals may in fact be due to an auditory startle-like reflex. So Sato and colleagues used wide field fluorescence microscopy of calcium signals in transgenic mice and found that ultrasound stimulation here over the visual cortex evoked activity in contralateral auditory regions, and that chemical deafening led to abolition of behavioural responses measured by EMG. Similar results were found by Guo and colleagues working in a guinea pig model, where cutting the auditory nerve removed the behavioural responses to ultrasound. Now, there are some questions about both of these studies, and certainly the existence of potential confounds does not, of course, mean there's no real effect of ultrasound. But it's important to control for them carefully in your experimental design. And to date, this hasn't really been the case in human experiments. So with that background, my colleagues and I decided to try and answer a couple of questions. First of all, we wanted to know whether the observed auditory confound may also apply to human studies, and if so, whether it can be controlled for. And secondly, whether transcranial ultrasound stimulation can be used to modulate higher order functions, and in our case, a higher order visual cortex in humans. So we conducted a couple of experiments to address these questions. First, a simple stimulation detection task with focused ultrasound applied to visual area V1. And secondly, a visual motion detection task with ultrasound applied to area MT using the fusiform face area as a control region. Prior to the main experiments, 16 healthy subjects underwent structural MRI scanning and functional localization of V1 MT, and FFA. Both acoustic and thermal modelling were carried out at an individual subject level to determine the optimal transducer placement and ultrasound amplitudes for our intended targets and safety constraints. So our structural images were segmented into grey and white matter, CSF, bone and scalp. And then using open source software called K-Wave, we modeled phase distributions back propagated through the skull from an ultrasound point source positioned at our intended target. So then subsequently, we did forward simulations to determine what the in situ pressure and thermal fields were. As long as everything was within established safety limits, this is where the transducer was placed, and if not, then we tried other positions. The transducer we used was this H107 Sonic Concepts with a base frequency of 500 kilohertz and a focal length of about 6 and a bit millimeters. And over here, this table shows the acoustic and thermal tissue properties that we used for the modeling, such as the speed of sound in the tissue, the attenuation in the tissue, the thermal conductivity of the tissue, and the specific heat capacity. Our ultrasound parameters that we used experimentally were determined by a maximum pressure of at the target of 0.6 megapascals and a maximum temperature rise in bone of 3 degrees centigrade. We experimentally verified our modeling procedure using CT images of an ex vivo skull, and then compared the modeling results against the actual pressure fields measured by putting this skull in a water tank and using a fiber optic hydrophone. As shown here on the right, the simulations on the top for V1, MT, and FFA closely matched the measured pressures shown here on the bottom. Although for the deeper FFA region, there was greater field fragmentation than we had predicted in the model, perhaps related to reflections from the petrous temporal bone, but also that the maximum pressures measured were lower than those modeled. So this slide illustrates our experimental setup. The participant is seated in front of a computer. The ultrasound transducer is held in place by a positioning arm and coupled to the scalp with a cone filled with degassed water that can be adjusted to vary the depth of the focal point inside the brain. The transducer and the participant's head are tracked by a camera and aligned to the previously acquired MRI scans via a brain site neuronavigation system. EEG is monitored continually throughout the experiment, and we cut a hole in the electrode cap to allow access for the transducer. So here are the stimulation, here's the stimulation protocol and the ultrasound parameters that we used. As I say, the ultrasound base frequency was 500 kilohertz, applied with a pulse repetition frequency of 1 kilohertz during a stimulation burst that lasted 300 milliseconds per trial. And the interstimulation or intertrial interval was 2.4 seconds. So for our first experiment, we delivered 100 trials of interleaved V1 stimulation or no stimulation, and asked participants simply to report whether or not they thought they had been stimulated on each trial. The mean D prime value of 3.6 shows that most could detect stimulation at well above chance level. Only one participant reported an occasional visual sensation akin to phosphenes. Instead, participants reported being able to detect a high-pitched tone on some trials, and they associated that sound with stimulation. The sound approximately matched when we asked them a 1 kilohertz tone, which was at the pulse repetition frequency. So what we did, therefore, was attempt to mask the auditory signal by playing a tone at 1 kilohertz through earphones on all trials. The mask began shortly before the ultrasound burst on stimulation trials and lasted for well beyond it at 700 milliseconds. When we did that, it resulted in chance level detection rates in all of the participants, except for 3 who were still able to hear the tone. But that then normalized when we increased the volume of the tone. Now, the EEG demonstrated an event-related potential over the CZ electrode associated with the unmasked ultrasound stimulation. and that had all the appearances of an auditory evoked potential. The masking tone had a slightly larger amplitude and earlier ERP when it came in. And when the ERPs for masked stimulation and sham trials were compared, there was no significance detected. We confirmed that this audible tone was due to bone conduction of a flexural wave at the pulse repetition frequency by placing a receiver near the auditory canal of our ex vivo skull, which was then coupled to the ultrasound transducer. A bone-conducted auditory signal was detected at the fundamental frequency, which in this experiment was 500 Hertz, and also at the higher order harmonics. But this was only when the ultrasound transducer was coupled to the skull, not when it was uncoupled. And also that signal was abolished when ultrasound was delivered not in pulses, here in blue, but as a continuous wave. The reason why there are two peaks either side of the fundamental frequency is not clear at the moment. We're trying to work that out. So I now turn to our second experiment, which was, as you recall, designed to establish whether TUS can be used to modulate higher order visual cortex. And our task was a straightforward motion detection task in which an array of randomly moving dots appeared either to the left or right of fixation, and the task was to determine whether or not the dots were on average moving to the left or to the right. And we first determined the percentage of dots, the threshold, a percentage of dots at which the participant was correct on 70% of trials. And then in the stimulation experiment, we sampled within one standard deviation of that threshold. Trials began with a variable fixation period. There was then the audio mask just prior to the presentation of the dots. Stimulation and no stimulation trials were interleaved in a randomized order, and the dots and ultrasound stimulation if present lasted for 300 milliseconds and were followed by a variable fixation period before the response period. There were exactly 270 trials in total, split across 3 blocks, and in order to determine the spatial specificity of TUS, we did the task twice, once with the stimulation directed at MT and once with it directed at FFA, counterbalanced across participants. So first of all, the stimulation detection results from both MT and FFA, when we used the audio mask with the ultrasound transducer at these sites, subjects were no better than chance, d' of 0, than detecting whether ultrasound was on or off. Looking at the accuracy rates for motion detection, we found here on the right a positive behavioural effect of ultrasound at MT. So movement detection accuracy was increased significantly by stimulation. In contrast, when applied to FFA, there was no difference in motion detection accuracy with and without ultrasound. If we look at the individual difference in accuracy with and without MT stimulation, we see a mean improvement of 4%, significantly above 0, and with just 3 participants who showed no or a negative change between conditions. The effect of MT stimulation was largely driven by behavioural facilitation in the left hemifield, i.e. the hemifield contralateral to stimulation, and a direct contrast of the TUS facilitation between MT and FFA conditions confirmed that MT-specific facilitation only occurred in the contralateral hemifield. Turning to the reaction time data normalized across individuals, here we find a significant site by stimulation interaction. Again, ultrasound to MT had a facilitatory effect on behavior, reducing reaction times. And this reduction in reaction times was significant for accurate trials, but not for inaccurate trials, suggesting that it reflects greater certainty with stimulation rather than some non-specific increase in motor speed. And once again, this effect was driven by the hemifield contralateral to stimulation, that's the left hemifield. So behaviorally, TUS to MT leads both to an increase in accuracy and a decrease in reaction time that's not present when stimulation is directed at the control site. Now, the EEG has turned out to be more complicated to analyse than we had hoped, in part due to the large auditory ERP that we see from the masking tone. So I'm going to hold back on presenting those results until we're more certain about them. With respect to safety considerations, no participant reported any adverse effect of the stimulation. Everyone underwent repeat MR imaging after the experiment, and structural images were visually compared by a clinical neuroradiologist to look for any evidence of change of damage, and none was found, including in susceptibility weighted images, which are especially sensitive to extravasated blood. So in conclusion, we have confirmed that TUS can be safely applied in humans. We've also demonstrated the need to mask for an important auditory confound in pulsed stimulation protocols, at least. We've furthermore shown that TUS to area MT results in improvement in visual motion detection, both with respect to accuracy and reaction times. So TUS, I think it shows great promise as a tool for non-invasive neuromodulation with unparalleled spatial resolution. And there's, lots of potential for development over the next few years, but there's still a lot of work to be done, I think, investigating its underlying mechanisms of action, certainly its safety and efficacy parameter space, and its potential for therapeutic applications. So I'd like to stop there, say thank you to my lab and our funders, and take any questions.